I'm Ingo Helbig, and I'm going to talk to you about STXBP1 disorders. And I think Tracy introduced STX, as we call this, as another DEE. What I would like to do during my presentation is to put some kind of context around this and introduce to you guys what we've done in STXBP1, which is a relatively new condition in the field that goes into clinical trial readiness. So here's an idea of why STXBP1 is so new. Martina used this term, the last 10 years, what the TSC team has built in the last 10 years. We haven't had the last 10 years. So STXBP1 was discovered 2 years after the TSC natural history study got started. And by the time that CDKL5 had their U01 started and looked at validity of the CCSA, we barely knew what our phenotypes were. Yeah, so we had a lot of catching up to do. And those are just two examples where STXBP1 kind of started out. It started out discovered as a gene for severe epilepsies. And then at the other hand, the last publications that delineated the broad spectrum talked about the non-epileptic myoclonus, the STXBP1 tremor, which is a very unusual subcortical myoclonus. STXBP1, to put this in context, is half as common as TSC, twice as common as CDD. Yeah, so it is a condition that is very underdiagnosed. For every kid with Dravet that's diagnosed, there are 2 STXs that don't have a diagnosis yet, mainly because the spectrum is so wide. So having said this, it is an ideal gene for drug development. So STXBP1 sits right there where neurotransmission happens. It is the master regulator of how our synaptic vesicles fuse in the presynaptic membrane and lead to basically the release of the synaptic content. It's the master modulator. If you knock this out on both alleles, there's zero synaptic transmission. It is a very clear biological target, and there are many trials and preclinical developments in place to target this mechanism specifically. It's not that far away from where Keppra works, where Briviact works, so STXBP1 is very, very targetable. But we have a problem. And this problem is that STXBP1 has a very broad spectrum. I think, Jenny, you just mentioned that the clinical spectrum is wide. I'd like to see in STXBP1 it's even wider. Yeah? So we have a very dynamic seizure course. We have epilepsy that happens in the first year of life is extremely variable. We have a tremor that we don't understand. We have developmental differences, and this is something that we needed to kind of figure out and understand. So we have subgroups that we're trying to make sense of. And I would like to give an idea on how we try to solve this big backlog, these kind of 10, 15, maybe 20 years of backlog by innovation, and how we were able to kind of fill this gap. So I took a photo earlier, and it says, "Be bold." So let me be bold. STXBP1, in our Simon's meeting, on the INSYNC meeting, which happens in New York every spring, is the rare condition with the fastest growing knowledge. Yeah, it is a very new condition in the kind of clinical trial readiness field, but we're kind of catching up rapidly, and we're trying to use new tools, new concepts, new innovation to make this happen. So first of all, disease concept models. First thing we had to do is kind of put lived experience into scientific terms. Qualitative research, this is data now from more than 1,500 minutes of interviews with patients and caregivers mapped to 3,600 references across different domains. And what came out of here is that we have this disease concept model. We can conceptualize lived experience, and lived experience kind of breaks down into symptoms impact, and the caregiver impact. And more importantly, and you have seen this during the kind of patient reports, these things change over time. Early on, it's seizures. Later on, we have to deal with developmental differences and behavior. When it comes to epilepsy trajectory, we had to understand how could we understand a disease that basically kind of changes before our eyes. And we came up with kind of this solution here. This is a so-called alluvial plot. And let me walk you through this. And there's always this kind of joke that pediatric practitioners understand this intuitively, adult neurologists, not so much. So let me try. Every bar is an individual. Every column is a month of life. First month of life, second month of life, third month of life. The colors indicate severity of seizures. The connectors in kind of blue or yellow show us changes. So at the very top, you have an individual who has many seizures per day in the first month of life and then becomes seizure-free. And you can imagine now how these different kind of patients weave in and out of seizures. And this is kind of an idea on how you can represent a dynamic seizure development across an entire cohort with what we call reconstructed data. So the interesting part here is that all these 37 individuals from our first paper in 2021, all of these individuals have had seizures at one point during this first year of life. But if you draw a line, there's not a single time when more than 50% of people had seizures. So this is how you can reflect and visualize a dynamic seizure course in one of these conditions that are very hard to grasp, because you can have infantile spasms, or some people don't. So let's talk about the natural history. Let's talk about the first year of life, and let's talk about what we do in terms of more advanced approaches, seizure forecasting and virtual trials. We collected all the information we have on STXBP1, more than 470 patients from the literature, from our centers, from collaborators. This is the variant scale. This is now all the phenotypes mapped to a standardized terminology, more than 360 potential phenotypes. And since we had no idea how to kind of show this and make this understandable, we chose these radar plots. It gives you an idea on how the different clinical subgroups in STXBP1 correlate and what the spectrum is. This is now kind of updated data that was just accepted for publication in Brain that looks at a reconstructed seizure history in more than 150 individuals across more than 1,200 patient years. Every individual has an assigned monthly seizure frequency and a seizure type. Yeah, what we can see here is that we now have a complete grasp of the seizure trajectory. We talked earlier about medications and how we think about medications. And one way is to look at trials. The other way is to look at what we call real-world data. You've heard this earlier from Anoop, that we want to look at real-world evidence and real-world data. And this is what it looks like. This is now a density plot of the different medications that we have. And we basically have reconstructed medication history for all our patients. And you can see there's a lot of Keppra in there, topiramate. Yeah? What we can now do is take the seizure data, take the medication data, and ask the question, which medication helps when? And this gives us these kind of plots here about comparative effectiveness. There's another angle to this. Once you kind of have this analysis, you have to ask the question, what does improvement actually mean? Now we have to move away from the typical trial assessment by asking the question, are we talking about long-term seizure freedom? Are we talking about seizure improvement? And we see some very clear patterns here. For example, phenobarbital helps with seizure reductions. That's not a big story, but we know this from the LGS world. It is often a honeymoon effect and doesn't lead to a longitudinal or to a long-term seizure outcome. This is where ONFI and the keto diet are more effective. We've done now machine learning to look at seizure forecasting. Can we now find some patterns in this first year of life that predict the group of patients that become refractory? Yeah? And we call this unpredictability. And we've taken this actually from the EEG world, where people think about seizure forecasting from EEGs, and we looked at the same types of methods. And we could point out patterns, and we know that the last few months in the first year of life are predictive of the later seizure freedom or not. Finally, we've taken the entire data and asked the question, we don't have trials yet, but should we be scared of this diversity, of this broad range of seizures where we have a single variant that may produce seizures in one patient, but not seizures in others? And is this potentially targetable? So we asked the hypothetical question, let's take all our reconstructed data and perform a virtual clinical trial. Let's pretend there is a medication that reduces seizures by 20%. When would we see this? And those are the two bars here. You can see we have a very high probability of a trial success between 6 months and 5 years, and then again between 7 years and 12 years, and everything in between is tricky because the variability of seizures is too high. This is how you can approach a rare disease where phenotypes change rapidly. And at the bottom you can see that we can break this down for the different seizure types that we were able to reconstruct. So, fastest growing knowledge. But seizures is more, or STX is more than just seizures, and we have to ask the question, what do we know about development? What do we know about gross motor scales? Here are reconstructions of milestones. This is taken from data jointly from our center and the Citizen Invitae database, where we were able to kind of really point towards milestones, towards achieved abilities in our patients, and we're able to really reconstruct this and go from 0 to basically having a full overview when our patients have achieved certain milestones. Here is data that is pre-natural history data. And this is data that we've taken from the medical records on standardized scales. So once you start looking in medical records, once you start looking at digital data that you kind of extract from the EMR, you find that there's actually quite a bit of standardized data in there. But it's not me putting this in there. It's our PTs or our occupational therapist or speech therapist. And here we're looking at initial data from a scale that's called GMF, which is a gross motor function scale. It's a very standardized scale in the PT world. And on the x-axis, you can see the age. On the y-axis, you can see the scores. 100% is kind of the scale of a typically developed adult that we would typically reach by the age of 5. And you can see this kind of cloud of points. So I see a little bit more in there. I see 2 parts. I see a first part on the left, when we have a broad range of developmental features. This explains, this shows why our patients are so different with developmental abilities early on. But I also see the second part, the green part, where we have some sort of like a convergence towards a trajectory that we can actually measure. This is where you can measure 10%. This is the kind of natural history we have to get at for outcome measures. Finally, we've now moved this on a national scale into a prospective natural history study. And Jim, who's kind of the scientific director of the X Foundation, I would just kind of tallied our numbers. We started in July. We have now 37 kids enrolled who've gone through an entire natural history protocol that involves developmental measures, and basically takes half a day per patient to really create this granular data that we need for clinical trials. So this is another thing. This is basically our recent acquisition. This is a quantitative EEG. This is one of the little STXs here who has this little cap on. And it's actually quite funny. So this is not staged. This kid was really happy because it took our techs maybe 20 seconds to get this EEG thing on. We call this the squid. It's this little kind of EEG net that sits in like a solution. You can take this out, put this on a head, no glue involved, and it's very quick. And it's mainly meant to kind of look at EEG background and biomarkers that we would like to develop. Yeah. And what I wanted to kind of give you during this kind of presentation is a brief idea how we can go from 0 to 100 in rare disease using some conceptual innovations and being creative about existing data, real-world data, and trying to put this into a larger scale. So I'm the PI of what is called STAR, which is the STX Natural History Study. We have 4 sites right now: CHOP, Colorado, Cornell, and Texas. We hope to add Stanford, Juliet, next year. It's on record now, yeah? And we have the kind of very detailed program, and we hope to enroll more than 100 patients in this year just to get this baseline data more granular for clinical trial development. So I want to thank my team at CHOP. I want to thank the STX Foundation and all the families. And yeah, I'll be on the panel later. Thank you.